welcome to another session of CEC Gurukul lecture series on plant reproductive biology. Continuing with our journey of pollen, we will be talking about the morphology of pollen in today's class. Pollen morphology is a very important aspect of studying pollen biology. Pollen can have different shapes. They can be circular, rhomboidal, triangular, rectangular, or of any other geometric shape. Then pollen grains, when they are liberated from the tetrad, that time they are dispersed as single units, but before that, when they are in tetrad, we can also know the polarity of the pollen grains. There are two poles in pollen grains, a proximal and a distal pole. It's just like we have two poles on the Earth's surface. The pole of the pollen grain, which is towards the center of the tetrad, is called as the proximal pole, and the pole opposite to that, that is the side which faces the outside of the tetrad, is the distal pole. Now there is one imaginary line which passes through the distal and the proximal pole. This is called as the polar axis. Now at right angles to this polar axis is the equatorial axis, which divides the pollen into two equal halves if it is an isopolar pollen. Equatorial axis is equivalent to our equator of the Earth. Now if we have to understand the polarity of the pollen grains, this can be understood only till the time the pollen grains are enveloped in a callose wall, that is in a tetrad. Once they are liberated from the callose wall, we cannot know the polarity of the pollen grains. There are two types of pollen grains based on the polarity. These are isopolar and heteropolar. In isopolar pollen grains, both the proximal and the distal poles are the same, whereas in heteropolar pollen grains, the proximal and the distal halves are different. So, in a very common language, I would like to say that isopolar pollens are spheroidal, and they have the same diameter in whichever manner you cut them, whereas in heteropolar grains the diameter could be in one orientation, the length could be more than the width of the pollen grains. In this particular slide you can see that the pollen grains, they can be shed at either a 2-celled stage or a 3-celled stage. Figure A shows 2-celled pollen grains of hippophae which have been stained with the bromine iodide, and 3-celled pollen grain in the second picture in Indotristica ramosissima, and these have been stained by ethidium bromide. Now all the pollen grains, irrespective of their shape and size, they have certain number of apertures on their surface. An aperture is a region of the pollen wall from where the pollen tube is going to emerge. So this is a region of the pollen wall where the exine is either very thin or it may be completely absent. It serves as a site for water uptake and, like I said, eventually pollen germination. So once the pollen germinates, the pollen tube emerges from this particular aperture. In most flowering plants, the apertures are typically lined by a thickened intine only, which means, like I said earlier, the intine is mostly absent in these areas. These apertures can be positioned at the poles, they can be present at the equator, or they may even be present all over the surface of the pollen grains. The number of the apertures in angiosperms is very variable. The number can be 0 or it can go up to 100. Now there are also certain species which lack these apertures, and such pollen grains where there is no pollen aperture present. Those are called as the inaperturate pollen grains as seen in Aristolochia species. So, if there is no aperture in a pollen grain, how do we expect the pollen grains to germinate? In these cases, the pollen tube emerges by breaking the wall, that is the exine wall, and then it comes out. The number of the apertures is variable and so is the type of the aperture. The apertures can be of different shapes and they can be of different sizes. Now, here I have listed certain types of apertures. We say that it is a pore when the aperture is circular in shape. It is called ulcus when the pore is present distally. That means pore is present on the distal end of the pollen grain, which is away from the center of the tetrad. Pentaporate pollen grains are pores which are present all over the surface of the pollen grain. Now, when the aperture is not circular and it is elongated in shape, we call it as colpus. Now, if the colpus is present on the distal end, then it is referred to as sulcus. Colporous is an aperture which combines features of both pores as well as colpus. Heteroaperturate pollen grains are the ones where there is occurrence of both colpi as well as pori in a single pollen grain, as is seen in Melistoma. Now, since we have to talk about the morphology of pollen grain, there is a very prevalent system called as the NPC system, which classifies the pollen grains on the basis of number, position and character of the apertures which are pollen on the surface of the pollen. So, this is called as the NPC system, referring to number, position and character of the pollen apertures. You can see that in the first horizontal row, there are pollen grains which are showed with different numbers of apertures. In the second one, we refer to the position of the pollen grains, and the third is the type of the aperture which is present on the pollen grains. So, it is the pollen aperture which is considered here in the system of classifying the pollen grains. Now, when we talk about the number, based on the number of apertures, the pollen can be monotrim, ditrim, tritrim, tetratrim, etc. So, that means if there is one aperture, that is, we say N1, it is called as monotrim. If there are two apertures, that is number is 2, then the pollen are referred to as ditrim. If the apertures are 3, that is N3, then the pollen is referred to as ditrim. If the number is 4, then it is tetratrim. If the number is 5, it is pentatrim, hexatrim for number 6, polytrim for N7 and anomotrim when the number of apertures is more than 7, and if there are no apertures, the pollen are referred to as atrim. So, this refers to the number of apertures, that is the N part of the NPC system. Then there is the position of these apertures. Based on the position of the pollen apertures, there are again 7 groups, starting with catatrim, that is the aperture is present on the proximal face, and this is referred to as P1. Now, if this aperture is on the opposite side, that is aperture is on the distal face, then it is called as anatrim and it is denoted as P3. If the apertures are present in both proximal and distal faces, then it is called as anacatatrim and we refer to it as P2. Another category is when these the apertures are present in the center. Okay, the equatorial region of the pollen grain contains the apertures and this is called as zonotrim. Then there is dizonotrim, like zonotrim, but it has 2 rows of apertures on the equatorial region. In pentotrim apertures are more or less uniformly distributed all over the surface of the pollen. And when we are not sure, we are not certain about the position of these apertures, then it is referred to as P0. The third part of this NPC system is the character of the apertures. Now, based on the different characters, there are different categories. C0 is when we are not sure about the character of the aperture. So, this is character unknown. C1 is when the aperture is like thin area, or it is also called as leptoma. C2 is pollen with 3 slit-like colpi. C3 is colpate, that is, it is like a colpus. C4 is porate. Porate is with a circular aperture or with a pore. C5 is colporate with both colpi and pore. C6 is pororate with a pore. So, we talk about this NPC system. NPC is now basically describing the morphology. So, the most important aspect of pollen morphology is the pollen shape, and I told you that the pollen can have different shapes. Okay. I would like to go back and show you one of the pictures which I showed you in the beginning when we were talking. Here on the right hand side you can see that there are different types of apertures which are present on the surface of the pollen. You can see panporate, tricolpate, tricolporate, that is, there is a colpus as well as a pore. Then you can see the apertures are present all over the surface of the pollen grains. So, it is important to understand the morphology of the pollen grains, and this study of the morphology of pollen grains is also called as palynology. Right. And then another important aspect of pollen morphology is pollen wall. Now, I would like to mention here that the land plants have been successful because they have a very great pollen wall which prevents them from all type of environmental stresses which they face on a day-to-day basis. Pollen wall is basically, it is a covering of the pollen grains, and this pollen wall is divided into an exine and an intine. Exine is the outer portion of the pollen wall. Let me tell you that pollen wall can be multi-layered. Okay, primarily when we talk about the pollen wall, we say that pollen wall is two-layered. The inner one is the intine and the outer one is the exine. Intine is not split further in most of the cases, whereas exine is divided into two more layers, that is the ectexine and the endexine. Endexine is just outside the intine, or we can say that is the inner layer of the exine, whereas ectexine is the outer layer of the exine. Now, this ectexine is also divided into three layers, that is tectum, baculum and the foot layer. Foot layer, as the name suggests, is the base of the exine, that is the three-layered ectexine. It starts from the foot layer, then there is a baculum which consists of rod-like structures, columnar structures, and then this is tectum. Tectum is basically formed when there are small beads or other structures present on the top of these columns, and they combine together to form another layer which is the tectum. Pollen wall is formed in two phases. In phase 1, the pollen wall develops during the tetrad stage, that is, the wall material is contributed by the cytoplasm of the spore itself, that is, the microspore cytoplasm is instrumental in the formation of the first phase of the pollen wall. Then there is phase 2. Now, phase 2 of pollen wall development takes place only once the microspores have been released from the tetrad, and this wall material comes from the tapetum, and most of the time this comes once the tapetum has degraded. Now this is a one particular picture in which you can see that there is this is pollen wall, and two very common terminologies are generally used. One is given by Erdmann and other is given by Faegre. Now there is a very slight difference, because in general the structure remains the same, with intine being the inner wall layer of the pollen grains, followed by the exine. Now you can see here that the grey portion represents the intine, that is the inner wall layer of the pollen, and on the outside of intine is the exine. Now exine is split into nexine and sexine by terminology given by Erdmann, whereas if we follow the terminology of Faegre, then the ectexine, like I told you earlier also, is divided into foot layer, which comprises the base of the exine, and then you can see these column-like structures which comprise the baculum, and of course where they meet together forms the tectum or the roof of these columni. Now beyond the tectum there are certain elements which are contributed again by the tectum. These are called as the supratectal elements, okay, and these are also a very important feature of the pollen wall. Intine surrounds the microspore cytoplasm and primarily is made up of cellulose and pectin. Now because of the presence of cellulose and pectin, intine is less elastic. Exine, that is the outer wall, is made up of sporopollenin. Sporopollenin imparts resistance to pollen against numerous environmental factors, and also sporopollenin provides elasticity to the pollen wall. Like I said, that intine is less elastic, exine is the one which imparts elasticity to the pollen wall. Pollen wall is generally highly ornamented and it shows vast diversity. Now it's the exine portion which shows this diversity, because intine is present inner to the exine. Exine has predetermined spaces or gaps, which we were just talking about a few minutes back, and these spaces or gaps are called as the pollen or the germinal apertures. These are called as germinal apertures because these are the places from where the pollen tube emerges. Now apart from intine and exine, that is the two wall layers of the pollen, there are pollen coat substances which are present outside the exine. They can also be present all over the exine, and they can also occupy spaces which are present on the surface of the exine. So these pollen coat substances are of three types, that is pollen kit, tryphene and pollen kit like structures. Now we will take these one by one. First of all, I would talk to you about pollen kit. Pollen kit is somewhat sticky, hydrophobic mixture of lipids, proteins, carotenoids, flavonoids and carbohydrates. Now because it is sticky, it binds the pollen together in aggregates, and this is very important to promote their collective dispersal. And when pollen are bound together, when they are present in groups, the visibility is more for the pollinators. And this also serves as a food source for the pollinators. Pollen kit prevents the pollen grains from desiccation. Because it is sticky in nature, this enables adhesion of pollen to the bodies of insects and even to other dispersal agents, because like I said, it has carbohydrates and lipids. It can serve as a reward for pollinators. Pollen coat substances, that is the pollen kit especially, it protects the pollen grains from the ill effects of the ultraviolet radiation. Proteins in pollen kit they are responsible for the pollen pistil interaction. That is, once the pollen lands on the surface of the stigma, there is pollen hydration, adhesion, and of course germination which happens, and pollen kit plays a major role in all these interactions, and it is also instrumental in the sporophytic incompatibility. That is, the sporophytic incompatibility is to an extent decided by the pollen kit which is present over the surface of the pollen outside the egg cyme. The other type of pollen coat substance is tryphene. Tryphene is a characteristic substance of family Brassicaceae. This is thick and highly viscous coating which is composed of a lipid and a proteinaceous fibrogranular component. These substances, this tryphene is synthesized by the cell organelles of the tapetum, endoplasmic reticulum, and the degenerating elaioplasts of the tapetum are instrumental in the production of tryphene. Like endoplasmic reticulum is responsible for the lipidic component, and the proteinaceous component comes from the elaioplasts. Sorry, I said it in the wrong way. A lipid comes from elaioplasts and proteins come from the endoplasmic reticulum. Then there are pollen kit-like structures. So these pollen kit-like structures, they look like pollen kit. Their function is similar to that of pollen kit, and these comprise of two components, that is the vicin threads and elastovicin. Now, vicin threads are non-viscous, non-sticky thread-like structures that help in aggregation of pollen grains. On the other hand, elastovicin is viscous and is also sticky in nature. Vicin threads, they originate from sporopollenin. They remain attached to egg cyme and they are acetylosis resistant. They are seen in Onegraceae, Ericaceae, and they are also performing most functions of protection and of binding the pollen grains in groups. They are seen in Asclepiadaceae and they are seen in Onegraceae. Then the pollen wall plays a very important role. What is the role of pollen wall? Regulates the pollen size. Now pollen size is regulated by the pollen wall. Pollen shape is also regulated by pollen wall, and pollen wall, because it protects the pollen from dehydration. If pollen they get dehydrated, then of course they will show shrinkage, right? And pollen wall is the one which controls this. Pollen wall also controls homeostasis of the cytoplasm, and it reduces fluctuations in the pollen volume due to variation in water content. Pollen wall is very important during dispersal when the pollen is exposed to air. Pollen wall controls the rehydration process with water coming from the stigma after pollen lands on the stigma. See, you would recollect from our earlier classes that pollen are shed in a dehydrated stage. When the anther dehisces and the pollen are shed, they are somewhat dehydrated and there is very little content of water in the pollen grains. Once the pollen lands on a compatible stigma, then obviously it absorbs water from the surface of the stigma, and then it increases in size. It's after this absorption of water that the pollen grain, it becomes metabolically active, and all these roles, these are controlled by the pollen wall. Pollen tube is emitted through the pores which are present on the egg cyme. Egg cyme is again the outer portion of the pollen wall. So, pollen tube will come out, it will emerge only through the apertures or the pores which are present on the surface of the egg cyme. The pattern and the distribution of these apertures also is determined by the pollen wall. Now, there is a very important fact here. The intine, that is the inner pollen wall, supports the hormomegathic process, or in keeping pollen cytoplasm hydrated during dispersal. Like I said, that pollen have very little, they have very little moisture, they are hardly hydrated at the time they are dispersed, they are more or less dehydrated. So that can lead to the shrinkage of the pollen wall, to the shrinkage of the pollen grain, and pollen wall is the one which prevents them. There is an important phenomenon called as hormomegathy, that is folding of aperturate pollen grains in response to change in humidity allows a partial dehydration of the cellular material while preventing complete desiccation and death. It's the ability of the pollen wall basically which prevents the pollen grains from dehydration. So pollen wall plays a very important role in the journey of pollen right from the time when the anther dehisces and till it reaches the receptive stigma. Okay. Now, quickly I would like to tell you that exine is the outer wall layer, intine is the inner wall layer. Exine is divided into ectexine, endexine, whereas intine is not further divided. Exine is composed of sporopollenin, and this intine is composed of cellulose, hemicellulose and pectin. Though exine is highly sculptured and ornamented, it is the one which provides pollen with distinctive features and is instrumental in studying the taxonomic details, whereas intine does not have any such features. Intine is contributed by microspore cytoplasm and exine is contributed by microspore cytoplasm as well as anther tapetum. Okay, and now the exine has one or more thin areas which are the pores, and there are no pores on the intine which are present. Now both have different types of proteins which are present in the germ pore regions. Okay, now these proteins can be used as a marker enzymes for uh, 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 if we want to separate the intine and the exine proteins. So we will continue in the next class, and thank you very much.